The overall goal of these procedures is to develop a strain of probiotic yeast that can be easily transformed to express and deliver heterologous protein to the murine gastrointestinal tract.
The main advantage of these techniques is that they allow for rapid generation of auxotrophic strains of probiotic yeast. These mutant yeast can then be genetically manipulated without relying on antibiotic selection. Oral gavage of mice and harvest of Pyrus patches also allows for verification of mutant yeast adherence to the gastrointestinal immune tissues. These methods will help answer key questions in the field of oral vaccination and facilitate experiments testing probiotics for the delivery of gastrointestinal therapeutics.
After preparing YPD medium and growing yeast overnight to saturation, dilute cells 1 to 10 in water in a plastic cuvette. Then, blank a spectrophotometer and determine the optical density of the overnight culture. With 20 milliliters of sterile distilled water, dilute the cells to a concentration of 10 to the 7th cells per milliliter. Then, pour the diluted cells into a sterile plastic petri dish, and with the lid removed, place the plate into a UV crosslinker. Next, expose the cell solution to cumulative incremental doses of UV irradiation, extracting 500 microliter aliquots following each